I'd like to begin this morning by first discussing the idea of synthetic biology. So synthetic biology means a lot of things to a lot of people. And the reason for this, I think, is that the practitioners of synthetic biology, the people actually using the technologies that we refer to under this umbrella, are very diverse from a multitude of backgrounds. Synthetic biology is very useful, and so we see it applied to a number of different areas. And this is extremely evident when we think about the users of this group of technologies. So, for example, people are using SynBio in biology. People with computer science backgrounds are picking up these techniques. Artists, students, teachers, really from across the board, these folks are applying these techniques in new and exciting ways. The technology is phenomenally accessible, and I think that's a big part of the reason we see it used. Okay, thanks. I'm sorry. So generally speaking, if you ask people for a definition of synthetic biology, you get very different answers back, depending on that individual's background, thanks, and the way in which they tend to apply those technologies in their own work. Excuse me. But what we as a community typically agree upon is that synthetic biology can be defined as the application of engineering principles to biological systems. This is kind of a textbook definition, and I feel like it really only gets us half of the way there. A better definition needs to think about the way we use synthetic biology to interact with the organisms we're trying to engineer. So synthetic biology is typically done through the genetic engineering, the direct and intentional genetic modification of organisms we are interested in introducing new behaviors in. And these organisms tend to be microbes, bacteria, yeast, and so on, but they can be higher organisms as well. The problem with the definition I just gave you is that it assumes that life, biological systems, behave like a circuit board, and this just isn't the case. Organisms are not machines, and we can't give them genetic directions and expect that they will interpret them exactly the way we want. Instead, this ends up being a little bit more of a conversation rather than us dictating some genetic change in behavior. We provide genetic instructions. The organism tells us in response, yes, I understand these instructions, or no, I don't. And then we need to iterate on this process and see if we can improve upon this relationship so we can coax the behavior out we're interested in. As a result, we can really engineer all day, but unless we think about how to talk to these organisms, we won't get what we want. For this reason, I would argue that synthetic biology has engineering intent, but it's much more well-positioned instead at the nexus of biology, art, and philosophy when we think about the way we have to relate to these systems and manipulate them toward these goals. The philosophy piece, I think, is particularly interesting in the appreciation of the ethics and the motivations driving a lot of synthetic biology technologies. So why might we want to engineer systems or these microbes in the first place? Well, as I've told you, this is an incredibly useful set of technologies, and there are a ton of different applications we can approach using synthetic biology tools. We have seen tremendous strides in synthetic biology applied toward human health and human performance enhancement. Synthetic biology has been at the basis of a lot of new gene therapy technologies that are being developed. Specifically, CRISPR-Cas, the genome editing tools, are being used there. We've seen synthetic biology-based sensors to detect pathogens in the environment or even in biological samples and clinical samples. We're seeing development of synthetic vaccines, new antimicrobial drugs, and a lot of other tools for promoting human health. Microbes can be induced using synthetic biology to produce a number of different compounds that are of great interest to us. One of those compounds is a biofuel, or several different varieties of biofuels that are currently under development and can be scaled up ultimately, leading to a really new, economical, and renewable resource for fuel. Other materials that we can produce includes polymers that can be used for textiles, small molecules that can be used as odorants or sweeteners. And then we see synthetic biology applied to agriculture as well, where it might be a little less expected. So an example there would be engineering crops to be more drought resistant or more disease resistant. We also see synthetic biology used frequently in bioremediation, and I'll talk more about that in a moment. But this is the use of engineered microbes to remove contaminants from environmental systems, environmental contaminants. Interestingly, these same tools that have really fundamentally changed the face of molecular biology research are being applied in very unexpected areas as well, and one of those is in art. So here in this image here, I'm showing a wreath that's been generated from bioluminescent bacteria. And I think this is a really interesting expression of the relationship between science and art that isn't always so obvious to us, but I think with synthetic biology in particular can be appreciated very directly. So the idea of genetic engineering is not a new one, but I do want to leave you with the impression of how synthetic biology has really changed the face of our ability to interact with our environment from a genetic perspective. For the last 10,000 years or more, people have been engaged directly in husbandry techniques that have let us go from the domestication of wolves to now all of the dog breeds we know and love today. This has taken many generations of selective breeding to go from the wild animal to the domesticated animals we know so well. In contrast, synthetic biology lets us really skip the millennia of breeding and all of the false starts and directly dive into the genetic code to coax out the traits we want. So how do we actually do that then? Well, DNA is the medium of synthetic biology. So DNA carries the genetic information for a cell. This information is organized into genes, which are functional units of DNA, that provide some instructions to the cell. So different genes confer different capabilities on different organisms. We can introduce genes, modify genes, or take genes away in order to manipulate the behavior of different organisms. Genes are organized within an organism on a structural unit of DNA called a chromosome, and organisms may have one or many, and all of the complement of chromosomes of genetic information in a cell is referred to as the genome. So most synthetic biology is done by manipulating genes at the DNA level, but we also have now genome engineering capabilities that allow us to actually manipulate chromosomes intact within the organism, and this has proven to be a really powerful technology. I like the language metaphor when thinking about how we interact with organisms using synthetic biology. So if we think about DNA as being composed of letters, in and of themselves, those letters aren't very meaningful, but when we begin to put them together in very specific orders, they form genes, again, these units of information that we can think of as being sentences. They're able to convey a very structured thought or a structured set of directions to the organism. These sentences then can be grouped into chromosomes that we can think of as chapters, collections of paragraphs, sentences, words, letters that are on a broader scale. And then finally, the genome can be thought of as a book, the entire set of directions for life for a given organism. And Sarah will use the cookbook metaphor moving forward. So what are my colleagues and I actually doing in this area? We have a very active synthetic biology group, and we're pursuing a number of different things in human health, in bioremediation, and elsewhere, but I want to tell you about our bioremediation application today. One problem we've been aware of is the presence of environmental contaminants that I mentioned before, and specifically tires as a major source of waste. So tires don't biodegrade, they form massive dumps, and the disposal of these produces an extraordinary amount of pollution. There is a recycling program, but it doesn't get us anywhere close to where we need to be. We had conceived of this as potentially a synthetic biology, somewhere we could apply a synthetic biology solution. From a materials perspective, it doesn't make sense to make biodegradable tires. You want these to be really robust and resilient. But what if we could find microbes that naturally degrade rubber and really synthetically enhance their capability to do that? And do that in a very controlled way, so we could implement a bioremediation process and potentially approach these massive tire dumps that are releasing all sorts of contaminants and major sources of pollution and drive them down using the synthetic biology approach. So how would we go about doing that? The first step we need to take is to identify organisms that are already engaged in this process and make them do it a little bit better. These organisms can be found by surveying microbial diversity, which microbes are phenomenally diverse. They have a tremendous level of genetic diversity that we can draw upon to begin to identify the relevant genes that some of these microbes might use in degrading these compounds. Once we find these genes in the organisms we're interested in, we can either enhance their performance in that organism or move them into a different organism altogether to complement some other capability we might want to draw on. After we engineer these genes and modify these proteins in ways that are helpful toward this end, we can think about how to implement this at a larger scale and again begin to approach this problem of how to degrade this compound. So we've had a lot of success in doing this, but one of the things that became immediately apparent to us was that we lacked the tools to talk to some of these less traditional organisms very clearly. Synthetic biology has really been built on the shoulders of standard laboratory organisms, and there's a reason for that. Quite simply, we know them very well, and we know how to talk to them in order to elicit a very predictable response. When we step out of the lab, when we start looking at these wild microbes, we don't necessarily have the same tools to communicate with them as cleanly. As a result, we really need to develop them in order to move this capability forward. So for example, even though, working back to this language metaphor again, even though the letters are often the same, the sentences might need to be a little bit different. The paragraphs might have to be conceived of a little bit differently. And we need the tools to think about how to generalize this approach so we can meaningfully interact with the microbial diversity and the genetic diversity that they contain as well. Excuse me, let me back up here. So Sarah Richardson has been and continues to be a staunch advocate for developing synthetic biology tools to really take advantage of the amazing biological diversity that's out in the world. Her pioneering work in this area comes at a key time to contribute to a shift in the synthetic biology community, where we begin to build the necessary skills to talk to these species and drive research forward in really interesting ways. I met Sarah while we were both graduate students at the Johns Hopkins School of Medicine. She was the one that was, of course, taking 3 times as many classes as usual from 4 different departments while still engaged in full-time research. As a Department of Energy Computational Science graduate fellow, she was tasked with learning how to straddle both computer science and molecular biology, acting as a bridge between disciplines that don't always collaborate well. She fervently believes Great. Wanted to make sure I wasn't doing that. She fervently believes both in science as a public resource and the importance of diversity in research and technology. Sarah has acted on this belief by becoming a fixture in Bay Area classrooms. Her recent L'Oreal Award was a celebration of both her science and her passionate outreach. You won't meet anyone who is more fascinated by bacteria or more excited by their potential than Sarah. She told me that her research goal was to be a germ wrangler, and I think she's well on her way. I do think she's just looking for the ultimate communication challenge. Learning to talk to bacteria might be a lot harder than getting computer scientists and biologists to see eye to eye. So with that, I'd like to turn over the stage to Sarah.
Hello. People ask me why I wanted to be a biologist, and I always tell them it's because I wanted to talk to aliens. And they're not sure how to take that, but it's 100% true. My favorite puzzle is first contact. How do you figure out what life is when you can't assume that it looks or thinks or talks anything like you? How do you open the channels of communication when you don't even know if it understands communication through the shuttering of airwaves? How do you, more importantly, make sure that when you do connect with it and it sees you and you see it, that you enter into a mutually beneficial symbiosis? So to mix some metaphors, how do we make sure we make friends with Vulcans and avoid the xenomorphs and the brain slugs? It's just such a cool problem. I'm practically able to accept that I am not going to be meeting extraterrestrials. I get it, I accept it. I'm not even that disappointed because the strangest life that I could possibly imagine meeting is here on Earth. It's right under our feet. Life has been trial and erroring its way across this planet for 4 billion years. And as a result, you find it in places that seem more like a punishment than a habitat. When I hear people say that climate change is going to make the Earth uninhabitable, unfortunately, I have to asterisk that for human life, because to quote the most famous synthetic biology movie ever, Jurassic Park, life finds a way. It finds a way. So I will share you one of my favorite little alien things. This is Myxococcus xanthus. It's an alpha proteobacteria. The picture on the right is what it looks like when you look at it under a microscope. It looks like tiny little trees. It looks like an alien life form. This thing is so cool. It swarms around the soil crust looking for other bacteria to eat. And if you want to catch it, you have to use rabbit poop. You put rabbit poop down. It has to be wild rabbit poop. It cannot be domestic rabbit poop. They don't like that. And it is just the most amazing organism. When it is done killing things and it comes back, it forms these stalks, and half of them die so that the other half can form spores and survive the dry times when there's nothing to eat. It is so well organized that people thought it must be a multicellular organism, but it's just one cell. It's very alien. It's very hard to talk to. We're not good at putting DNA into it to get it to learn tricks. This is one of my targets. I want to wrangle this thing. It'll be amazing. Because it has to form these structures, it has these chemicals it uses to talk to other members of its species. It turns out a lot of those chemicals are very potent anti-cancer drugs. There is a reason beyond just, whoa, it's cool and it grows in poop, to get after this bacteria. It's figured things out, and that's the common thread between all of the life forms on Earth, is when it figured it out, it wrote it into its DNA. We all share DNA, even phages, viruses that affect bacteria or viruses in general. People tend to say that's not life, but they use DNA, which means they can store recipes, they can store genes. The phage has a small genome. Myxococcus xanthus, that amazing soil bacteria, it has a bigger genome. It's actually the biggest genome we know about in bacteria, about 9 million base pairs. C. elegans, which is a nematode, a little worm, it has about 100 million bases of its DNA. It's a lot of DNA to look at, but only 20,000 genes. And here's the scandal. When we sequenced the human genome, we found out we actually have fewer genes than that little worm. People were shocked. That is one thing humans can't brag about, is how many genes we have. It turns out we use them better. We can pat ourselves on the back for that. That's why we can get away with 17,000. But then the pine tree, 50,000 genes and 22 billion bases. These are huge, huge amounts of information that help us distinguish each other. This is why we're all different and yet also similar. DNA is the common thread. I don't really blame people for being a little boggled by evolution. I'm boggled by it. I can't imagine the timescales involved, because I have trouble remembering that the 90s were 2 decades ago. Let alone that 4 billion years of life has been happening. But as a result, evolution has kept the genes that help us survive. So written it into our DNA, the genome. Consider it, like Jessica said, I consider it a cookbook, because I get to look through it and see, ooh, this is a good recipe. This is a gene that helps this organism survive in a little hellhole where it's just growing on sulfur. Well, maybe I need to get rid of some sulfur over here. This is a recipe for how you can cope with sulfur. I think of genomes as cookbooks, and they're living cookbooks because they change. Your genome was passed down to you very carefully and tenderly by your parents with small alterations, an extra pinch of salt here, an extra reflex there. The changes are what help us differentiate from each other and what help dinosaurs turn into birds, even though it's very hard to imagine. They're sharing the legacy of a genome. And when you put them all together, what we have is a library. The planet is a living library. That's what I should have called my talk, not a genome is a cookbook, but the planet is a library. We have to preserve the diversity of our planet, because 4 billion years have given us recipes that we are not capable of inventing ourselves. Some of the things that life does is still so puzzling to us that we do not have an explanation for how it works. It just works. It's a great time to be in biology because now we have even better tools to look at the real freaks out there that are doing freaky things and break it down into action. The planet is a library and we have to preserve it. And it's a good note to also realize that no organism is an island. The other thing about genomes being these libraries is they're not individual. Everything on this planet walks around with an entourage of bacteria, and the bacteria have their entourage of phage, which are one of the most abundant organisms on the planet. There's just, the numbers are staggering. Nothing is an island, and when you start putting genomes together, they affect each other. The original genetic engineering is just cohabitation. That's it. You move in with your spouse and you start to change your behavior a little bit because now you have to cohabit. That's every organism on the planet. When their ecosystems start to overlap, they start to change, and those changes are reflected in the DNA. So the original genetic engineering project is agriculture. And before you start patting yourselves on the back about that, we didn't invent it. Those termites in the top left corner, we think that 30 million years ago, they fixed this fungus. They made a monoculture of it, and they tend to it. They select the ones they like. Luckily, I think their patent has expired after 30 million years, so if we want access to it, we can get it. That damselfish farms fungus on coral reefs. It'll actually pluck coral out so it can put an invasive fungus that it likes to eat better in there. Does that sound familiar? It sounds very much like something a human would do. Ants in the bottom are famous for marine fungus and aphids, just like little cows. And recently, scientists have observed monkeys in Japan feeding deer and then riding them around like little antlered Ubers. So when we do it, we call it domestication. When they do it, we call it symbiosis. But I want you to remember that it's the same thing. They're changing the genomes of everything in their environment to suit themselves. They're doing it. They're doing genetic engineering. They're just doing it the slow way. And we're pretty good at doing it the slow way too. All of these are one species. But when you select over generations for a feature, you get Brussels sprouts, or you get broccoli, or you get kale. These are all exactly the same species. This was genetic engineering done before we could see the changes in the DNA. But now we can. We can look at these changes, and we've added to our library by doing this. Now we can go back and say, oh, this is the gene that we were actually going for when we made Brussels sprouts. Delicious. And actually in the bottom right, broccolini is a very recent hybrid. It was invented in the 90s by mixing Guilan and broccoli. So we're still doing this. And of course, you're familiar with barnyard animals, the domestication, the species we're so proud of subduing that we teach our children to be proud of it. Like, what sound does that subjugated animal make? I include the cat in here as an argument piece. I'm totally willing to argue whether or not cats are actually domesticated or if they domesticated us. It fits into, we're changing each other. We're changing each other. But agriculture has always meant the husbandry and the creation of genetically modified organisms. There's not a lot of point in feeding something or taking it into your home if it's not doing something for you, and if it doesn't change its behavior to make sure that it fits well in your home. We've also done this with microbes. And I think we need to be more aware of the microbes that we have domesticated and that we use every day. Genetically modified organisms make all of the antibiotics that we give ourselves as medicine. We have genetically modified, even before we were doing GMO with synthetic biology, yeast to give us delicious beer, bread if you're under 21. Cheese is a symbiosis project. It's not possible to know if the bacteria that helps us make cheese came from our mouths or came from cheese and now is happily living in our mouths. But that bacteria, its closest relative is the first bacteria to colonize our bodies when we're born. That's how close we are to cheese. I didn't know xanthan gum was entirely made in bacteria, so that was a new one on me. I did know that MSG, entirely made in bacteria. These are products that are the world over and 100% made by bacteria that we found in the wild and said, you're good at that and I like how it tastes. That's genetic engineering in a nutshell. But it's a slow way to go if you can only breed. It's a slow way to go. And it's indirect, and therefore you can bring along problems. Dalmatians are beautiful, and they're going deaf. German Shepherds are loyal and smart, and they're having hip problems. Golden Retrievers are Golden Retrievers, and they have cancer problems. I would like to think that if I now, especially having learned from these mistakes, being able to see the gene changes that we wrought slowly, carefully, or indirectly without even a forethought over 20,000 years to these animals, I might be able to go back to the wolf, put the changes in directly, and skip the hip dysplasia. Because the genes are connected in that thread of a genome, that one big molecule. And when I picked one to accentuate by selective breeding, I had no choice but to bring in unintended consequences. The new tools that we are advocating for genetic engineering, they offer us the ability to maybe skip some of these unintended consequences that we can now see. So some recipes are also harder to access than others. We are not good, like Jessica said, at talking to bacteria. These are all pictures of bacteria. The top left one, those are magnets. This bacteria biomineralizes and makes its own tiny, perfect little magnets. It uses them to align itself to the Earth's magnetic field. I don't know why. I don't know what it needs to do. It doesn't have Google Maps. It's not trying to get to the closest Starbucks, but it could if it wanted to because it knows which way north is. It makes its own magnets so perfect that we can't even compete. We actually harvest some of those magnets. The next bacteria to it is Animax. It grows in the bottom of wastewater. And so it has a very ammonia-rich lifestyle, which is, like I said, more of a punishment than a habitat. The way it copes with this is to turn that ammonia into something it can eat, partially by turning it into hydrazine, literally jet fuel. This bacteria makes jet fuel. And then to save itself from the jet fuel, which is super toxic, like you would expect, it makes these high energy molecules called laterines. There's one at the bottom there. Basically looks like carbon stacked in boxes. That's a really weird molecule. Nobody thought life could make that. And it makes it to protect itself from the other thing nobody thought life could make. The next one in our cavalcade of amazing bacteria, the little grids on the left, those are gas vesicles. These bacteria can control their buoyancy in the water column. So they can control how deep they are in water. In response to things like oxygen saturation or UV light, they can move themselves up and down to get better places. They use it to, guys, it's just stunning. The bacteria on the right is making something called an S layer. It uses proteins to make this tiny little mesh that you could conceivably attach things to. It uses it to protect itself, to move around. It's a perfect mesh. This picture is a scanning electron microscopy. Nanoscientists who don't even know biology are trying to get into biology so they can figure out how to make things this perfect. We're learning from these things. Bottom right almost seems mundane at this point. This bacteria turns sunlight into sugar. Oh, it's not magnets. The bacteria on the left, those extracellular things sticking out of it, those are called cellulosomes. They're extracellular machinery it makes and puts out, like little worker drones that break down cellulose. It made its own drones, guys. We cannot compete. But the best thing that bacteria have made, in my opinion, and the reason we're talking about this, because this is something we need to discuss, the only reason we are even talking about accelerating our genetic engineering or being better at it is because we stole another tool from bacteria, the one that actually does the DNA editing. We did not invent that. We watched it happen because we were observing our library. Sometimes to make biology work, you just have to get out there and sit in the library and look around for a bit. We have never invented anything that we genetically engineer. We saw it in the wild somewhere. And we either accentuated it there or we borrowed pieces of it to put someplace else, but we have never made anything unnatural. It's all based in stuff we saw. And that includes the genetic editing technologies that we're using now. They were all made by bacteria. They had a head start on us. So I want you to picture this. We're living it. Novel DNA technology is roiling the field. There are calls for a moratorium. There's calls for action to regulate it. People are worried that we are going to edit humans and we're going to change things that's going to get out into the ecosystem and wreak havoc. There's threats of Nobel Prizes. There are the big people come together and they form a meeting and we're talking about what this is allowing. What year is it? Is it 1975 or 2015? Part of why I want to put this in context as genetic engineering as a new step, not a huge one, but a logical next step in our progress of relating with other animals and life on the planet, is we have been having this conversation for a long time. This is not a new conversation for us to have, and it's very good for us to continue to have it, but also to not forget that we have been having it so we can capitalize on the progress we made before. In the 70s, they had suddenly gained the ability to cut DNA wherever they wanted and then stick it back together. They did not have the ability to do it in a cell, and I'll show you the differences, but it caused people to ask, what are we going to do with this? Which is a great question. It caused people to ask, are we going to change human DNA with this? They ended up not really doing that because it's hard. And now we're facing the same thing with CRISPR-Cas9. It appears to have come out of nowhere. I'll show you it didn't. It's a bacterial tool. And we have now the capability to maybe ask ourselves technological questions that we couldn't ask before. So it's very important that we discuss this. But I wanted to give you guys a little bit of context too for how normal CRISPR is. Like I said, phages are the most common organism on the planet. Viruses on this planet, but we should not think of them as antagonistic. They're not, most of them are not pathogenic. They don't, and I don't even mean, oh, they didn't mean to kill you. No, most of them are not lethal. Most of them are actually necessary. If the planet is a cookbook, viruses are like the reserve section. They are just storing information for us, and periodically they offer it to you. And you're like, no thanks, I don't need that right now. And they're like, no, really? You want some of this? Because honestly, they are relentless salespeople. So a virus's MO is to come and knock on the door and be like, you want some DNA? It's good DNA, it's got some stuff in it you want. And you don't always want that. If you listen to the virus and didn't regulate it at all, it's going to ask you to make more and send it out to spread the good word. I was so helpful to you, you let me in, let's go spread the word. The reason cells have defenses against this is because that's a big use of resources to take on a virus's agenda. Maybe I don't want to do that. So the cells developed what you should think of as regulatory mechanisms to sort of slow the virus's roll. And we call them immune systems because they remind us very much of how we fight off viruses. But because the bacteria are forced to interact with viruses at the DNA level, it turns out their methods are our tools for editing DNA. This is exactly how we do it. So the first one that caused the big discussion in the 70s, they're called restriction enzymes. And the way they work is the bacteria noticed there are some sections in this foreign DNA that have a sequence that we can recognize, and I can make a protein that recognizes them, and when it sees them, it cuts them. Great, we harvest these restriction enzymes out of bacteria and use them to cut DNA all the time. We don't get them to work inside cells, we get them to work outside cells, so it's more the way we synthesize things outside. But 90% of the bacteria on the planet have these systems. So every bacteria we meet, we're like, ooh, new restriction enzymes, new ways to cut DNA. You might notice a logical problem here. How does the cell avoid cutting its own DNA? Because if it only recognizes 6 bases, that means you're going to have that motif at random every 4,000 or so bases, or 5,000, yeah, about every 4,000 bases. So another really cool trick they did, they said, well, okay, so we're full of that too, and we can't have our own proteins cutting our own DNA. They actually modify, they have another protein that recognizes the same exact spot and tags the DNA. And we steal those proteins too, those are great. We use both of those. This is entirely a bacterial system. This is the technology that was the CRISPR of the 70s. And now it is not considered dangerous at all. It is considered just a tool, it's a screwdriver in our toolkit. But that's because we were very careful about how we used it, and it also has very stiff limitations. It does not work in live cells. We can't use it directly in live cells, so it's a tool that has its limitations. CRISPRs also regulate foreign DNA. They do it somewhat more cleverly. It's more difficult, and that's why only 40% of bacteria actually have CRISPRs. But I guarantee you, you all have CRISPRs in your gut because the bacteria in your gut have CRISPRs. They're running right now helping protect your gut bacteria from the relentless salespeople that are also in your gut. You're an ecosystem unto yourselves. You should pat yourselves on the back for maintaining it. So CRISPRs also regulate foreign DNA. They do it a little differently. They recognize a motif in the virus, and they write it into themselves. They write it into their own genome so they can actually pass their immunity to this virus onto their children, which is, again, a trick I wish we knew, right? Because I would get a chickenpox vaccine and my kids wouldn't have to. That would be a pretty cool trick. Bacteria figured it out. Once they've written it into their genome, they can program a protein to go get exactly that spot. So of course, the phage change, and you have to pick a new spot, and the bacteria write that new spot in and go after it, and they can go get that. And then the bacteria picks a new spot, and they write it into their genome, and the bacteria has to go get that. And when we saw this happening, and the first CRISPR was spotted in 1989, we saw CRISPRs happening. We didn't even know what was happening. What is happening? What is this for? They're very easy to spot in the DNA because they're repetitive. What is happening? And then they realized in this decade, oh, we can just take the CRISPR and that protein and have it cut DNA. That's the big technology that they're fighting about now. They stole it from bacteria. They say we can actually write a target for this protein and have it cut the DNA we want. There are limitations. There are limitations. For one, this system evolved in bacteria, and remember, bacteria have smaller genomes than humans do. So the amount of space, amount of bacteria that this protein can recognize properly is only about 30 base pairs, which is enough for it to be unique in even the biggest bacterial genome. When you have 10 million bases, 30 base pairs is a pretty good unique tag. When you have 3 billion in a human genome, it's not very unique. That's why CRISPR is very hard to use in human cells. It's actually, there's a lot of reasons, but that's a big one. It's not very accurate. So I really hate this stuff, I'm not gonna lie. Every single time they talk about designer babies, they have a white baby on the cover. It's, and that is some of the implication, that if we can now engineer the human race, we're gonna make sure that we make beautiful, healthy, blonde, blue-eyed babies because they will have a good life. They'll be healthy and they'll also be beautiful. There's so much baked into these covers that I think, for me, it overshadows the awesomeness of the technology involved. This is not the best use of our technology. And also, like I said, it's very hard to do. It doesn't work very well. CRISPR is not the thing we're going to use in this current form to edit people. This conversation is important to have, but what I want you to be excited about is the potential for CRISPR in bacteria, where it works really well, where it's already working to solve some of the problems we've made for ourselves that are much more pressing and affect many, many more people than designer babies. That problems we can solve with genetic engineering. So my genetic engineering recipes for the future are fertilizer-less agriculture. This is the perennial algae bloom, toxic algae bloom in Lake Erie. These toxic algae blooms are often, it's impossible to exactly say exactly what, but we're pretty sure. Fertilizer runoff, phosphate, iron, nitrogen that we pour onto plants to get the plants to grow big and tall and have lots of them so we can eat a lot. They can't absorb all of that in that form. It's a very crude and ham-handed way to do agriculture, is to just add these things to the soil. What happens is they run off, they end up in the watershed, and algae go nuts. We're fertilizing algae, and these algae happen to make toxins that are bad for the fish, they're bad for us. They choke out the oxygen in the water, the fish can't breathe. Plants are traditionally fertilized by their buddy bacteria. When we start adding nitrogen to the soil, we are allowing the plants to quit tending their relationship with their buddy bacteria. And they do more things than provide nitrogen. They provide plant hormones, they help keep pests away. We totally missed that. We started adding nitrogen, the plants got big, the bacteria started dying off, we didn't notice. Now the plants are getting smaller because the bacteria aren't there anymore. One of the things we need to do with genetic engineering is buddy up with the plant buddies, put them back, and help us reduce our dependence on added phosphate, added nitrogen, added iron to these environments, and let the bacteria go back to doing it and tweak the bacteria so they do it even better. and not have this happen anymore. Fertilizerless agriculture, genetic engineering recipe for the future. Heavy metal bioremediation. This is another problem we have created. We've toxified some of our environment with heavy metals. Luckily, bacteria, there are some bacteria out there, I didn't include this on the amazing magnet slide, some of them pull heavy metals from the water. We're not very good at talking to those bacteria. We haven't domesticated them. We haven't harnessed them for this use, but it's possible that we could make renewable, genetically modified organisms that help pull heavy metals from the water. We need to look at that. Greenhouse gas reduction. Greenhouse gases are caused by our agriculture, but the more proximal cause is gut bacteria. We've all farted, you know. You fart because you have gut bacteria that are healthy. Farts are great. They're great, they're a good sign. But we might be able to tweak our bacteria so that they redirect that energy in a less juvenile direction. This would have a huge impact on our planet, to be able to talk to the bacteria that are producing methane and get them to redirect it. We can do this with genetic engineering. We can try this. Novel antibiotics, all of the antibiotics and a lot of the medicines that we use, because they have to work on cells, we steal from cells, we borrow them. We can't talk to, we're not able to directly manipulate a lot of the cells that make these kinds of antibiotics. As an example, erythromycin, made in Saccharopolysporia erythraea, is the only place we get erythromycin. We just grow it up in vats. I think they've done, you just hope you get a good strain that's like, oh, even better erythromycin. But if we can do genetic engineering, we could get into that bacteria and tweak the erythromycin a little bit, maybe make a new antibiotic that the other stuff hasn't seen before, and continue the effectiveness of some of our antibiotics if we can get directly into the bacteria. These are all genetic engineering recipes that offer to have a much bigger impact for our planet than designer babies. These are things that, rich or poor, no matter where you live, you need clean air, you need clean water, you need food, and you need medicine, and we can make that with bacteria. So the planet is a library. It has a lot of the solutions we need for the future. They're in there. We have to be open to it. We have to be preserving our diversity, accepting that it's changing, that it's a very act of living with it. Everything's changing. We respect that. This is the scientific reason you should have diverse boardrooms. There's no monoculture on the planet. That's a sign to me that monoculture is not a good idea. You need diversity. You need that backup of other people thinking other things and other organisms specializing in other things. This is how we're going to survive. Yeah, that's it.
Thank you so much, Sarah. Your enthusiasm for biology is absolutely infectious.
I see what you did there.
Yeah, let's give it up for Sarah and biology. I'm Eliza Barkley. I'm the Science and Health Editor at Vox.com. We've already got a lot of questions, which is awesome. We will have a couple minutes for them. If you haven't asked one or you want to vote, go to Slido.com or go to the South by Southwest Eco app. First, I have a couple questions. So I'm curious, what do you think are potential risks for some of these applications that you're really excited about, like the fertilizerless agriculture? And is there something about CRISPR-Cas9 where you can guarantee control over this process?
No, I can't. I wish I could guarantee control, because I know that would make a lot of people, well, no, you wouldn't trust me anyway. No, I can't guarantee control. One of the differences, I think, between engineering and bioengineering is that engineers have a very low tolerance for the failure, and their components don't change themselves out from under them. So transistors don't make imperfect copies of transistors, but cells make imperfect copies. So part of bioengineering is being tolerant of its ability to change itself in response to what you asked it to do. So the trick is not going to be to come in dictator-like and say, now you are going to do this. It's going to be the same thing that we did with domestication. The dog is already kind of hanging around, the cat's hanging around. You don't ask a goat to go catch mice for you. The cat's so much closer and so much better at it, and so much easier to control when you're letting it do stuff that it already kind of wants to do. So the best thing I can offer for you is not perfect control, but foresight and attention to context in what I'm doing. I don't want to be a dictator. I want to be a symbiont. I want it to want to do what I want it to do, And that's where I'm going to get my control. More like a germ wrangler or a dog whisperer than a dictator.
Cool. So how easy is it to get access to CRISPR-Cas9? I mean, can you order this off the internet? And what does this mean for citizen scientists who might want to use it, or for people who might have like nefarious intentions with this technology?
I think if you have nefarious intentions, it's a lot easier to not use CRISPR. One of the cool things about it is it is very available. That's one of the amazing things about biology, is that people have always been able to breed their own cat. You have access to the animals, to the organisms. You can do it. They make some pretty weird looking cats. But to get it to do what you want it to do is a challenge. You can get the CRISPR. Can you use the CRISPR to make something? Even I have trouble with that. It's still a very raw field. There's going to be a lot of trial and error, but if you're actually bent on ill intent, you can go get pathogens from the wild. It's a lot easier to take a pathogen like this than to make one with CRISPR. It's not that cooperative.
So you don't imagine sort of citizen scientists out there using CRISPR to
I do imagine them using CRISPR, but not to make pathogens. No, I think whatever they're going to try to do with it, it's going to be hard. It's the good stuff, the bad stuff, it's all going to be hard. But I like that it's democratically available. It does increase the access of people who might not have traditional access to science for no fault of their own. And when we talk about only scientists should have access to it, at this point in time, we are actually talking about privileged white men having access to it. And I'm a little sensitive to that because my students always ask me, you don't look like a scientist. When is the scientist getting here? Which is an annoying question, but I let them ask it because then I get to ask, what does a scientist look like? Mm-hmm. What are you thinking when you say, should only scientists have access to it, or who are the citizen scientists if they're not the scientists? The students always give me the same answer. It's a white guy with glasses, a lab coat, and no social skills. I did not make that up. That is what they tell me every time. So when we say, will everyone have access to this? At this point in time in our society, yeah, this is a way for people who might not have access to science through just institutional shapes to get access to it.
So do you think that fears around GMO food, which we know are abundant out there, and also fears around the implications of the human embryo editing are going to inhibit the spread of this technology going forward?
I think it's right that we discuss it and then we take it slow. People's feelings in terms of human genome editing, they're worth taking into consideration and slowing down for. It is worth making sure, especially because taxpayers foot the bill for our science. We need to make sure our taxpayers are on board for this. So yes, I want to talk to people. I want to hear their concerns. I want to know what they think about GMOs and why they think that, and especially human GMOs, yes. I think inhibit is not necessarily the word I tend to think of it as regulating. If people are scared and I'm not doing a good enough job communicating why I'm not scared. And that's on me.
Do you think this technology needs to be regulated going forward?
Regulations are generally good.
And you've told me that you think that actually the potential for using this technology in human health is more so in the human cell lines as opposed to the embryos.
I would like to not throw out the baby with the bathwater and say no CRISPR because we could make designer babies. When in cell lines, when you're studying cancer and you're looking for cancer treatments or HIV treatments, that the use of this technology in human cell lines has no risk of creating an embryo that is an ethical problem. I think that is an area we should encourage people to think about allowing and promoting, yeah.
Okay, we're going to take a couple of the top audience questions. Okay, can similar bacteria that degrade rubber tires also consume the huge plastic islands growing in our oceans? Yeah.
It's happening. Even when there was oil spill, they found out, there's a bunch of bacteria in the ocean that are capable of breaking down oil. The genetic engineering trick would be to help them do it faster so that we don't have to wait 20,000 years for it to disappear. Yeah. So yes, that is a great genetic engineering recipe, is get rid of the plastic or make plastic that is actually biodegradable, yeah.
Okay, why do you think GMO seeds have gotten a negative response from consumers when the science supports using them?
I think that's communication and the struggle between moneyed interests and the perception of why people are selling something. I don't know. We need to do a better job communicating how they were made and why we want to use them, and also to remind that there are risks and there are rewards, and we can't just focus on the rewards and we can't just focus on the risks. We have to balance them, and especially in these times when climate change is changing our planet, selective breeding is not going to go fast enough for us to continue eating the amount we eat and the stuff we eat.
And just to follow up, I mean, who do you think is responsible then for communicating this, for the education? Is it scientists? Is it the government? I mean, clearly there are advocacy groups that make strong cases against GMOs, but who's the best voice to sort of talk about the science on the other side?
Well, scientists and government, most science is funded by tax dollars. I feel personally responsible when I'm taking taxpayer money to make sure that taxpayers know what I'm doing and why, not least because they can then tell the government to keep giving me the money to do it. Like that one right there, give her the money, all the money. So I think it's all of our responsibility to participate in the debate. We are taxpayers, we have a stake in this planet just by dint of living here. It's up to all of us to ask. If we didn't explain it properly, make us explain it properly. Don't let us dismiss you. If you don't like the answers, that's totally fair, but make sure we explain it, ask us, and the scientists need to say, and the advocacy groups need to advocate. I think we're doing a pretty good job of communicating. I think it could always improve, but it's not the worst. It's not.
One quick follow-up. Is the government currently funding these environmental applications of CRISPR-Cas9 that you mentioned?
Basic science research is difficult to directly link to applications. So I would say, yes, we're doing all the things we need to do to lay the groundwork to then be able to launch into application. And there are groups that have even better ideas than I do, and they're closer to the work, and just amazing things. The scientific community is very brilliant, very creative. So I would say, yeah, we're working on it, but we're not going to hide anything from you. You'll know when it's happening.
You talk about the benefits of diversity. Doesn't genetic engineering reduce diversity and the potential of diversity?
No, I'm not capable, nobody is capable of putting out an organism that doesn't change. Diversity happens as soon as I put the bacteria on a plate to grow and it starts to divide. It is no longer a monoculture, even in itself. There's individuals, it's a whole population of bacteria that are different. When it goes out, and if it goes out into the environment and starts to interact with the other life forms, they are just going to shift. Even invasive species, we think of them as destroying diversity. What they're doing is changing it. They bring along other bacteria, they bring along other organisms. It's tough to think about. We tend to think this is how it should be, and this is, but we've changed our ecosystem so much. It's a little easier to think of it as changes that have a net positive good and changes that have a net negative, but we're not going to avoid changing. And no, it's not reducing diversity, it's just changing it.
Okay, last question. After all the research you've done on life, do you believe in God?
Oh man, that's the hard ball. I am in awe of the planet. It is, like I said, I can't even, you ever lay awake at night? How do they do that? How does that bacteria make magnets? Seriously. I would rather, I don't know, I like the fact that science and faith don't have to collide. I am not going to tell anybody else that there's no God, because look at this planet, man, it's amazing. We can coexist on that, but no, I don't believe in God. But I don't blame you if you do, because look at this planet.
Well, it actually looks like we have 5.5 more minutes left, so we're going to keep going through these questions. They are really good. And if anyone says we need to stop, we will. Okay. Are there any Jurassic Park-esque risks to this? How do we ensure a T-Rex microbe doesn't go rogue?
That reminds me, there's actually a bacteria that eats other bacteria like a vampire. It swims, it finds the one, it attaches itself, and then sucks out the other bacteria's juices. So I like to tell the kids, vampires are real if you're a bacteria. How do we ensure a T-Rex microbe doesn't go rogue? Part of it is that when we go into the lab and we engineer a bacteria, it gets really good at living in the lab. Almost accidentally, we don't on purpose make it good at living in the lab, but it takes a lot of cues and it gets good at living in the lab. Going rogue involves that it doesn't stop being good at the things that would allow it to go rogue. There's not a lot of rogue stuff out there now. And so when it gets out and tries to go rogue, there's a bunch of other stuff that's like, you're not from around here, are you? So some of ensuring that it doesn't go rogue, though, is domestication. Now that we have the ability to directly edit DNA, we can take away bits of its toolkit that it doesn't need in the wild or in its application. I want to be very careful about just assuming that I'm putting stuff in the wild. Even in the lab, I would like to be able to take away deliberately, to reduce, not to add things that might change later, but to take away. It's a lot harder for an organism to gain a gene than it is for it to adapt or to change a gene. So when I take things away from it, it's a lot like taking away my golden retriever's ability to go hunt squirrels for himself. If I leave my golden retriever in the backyard, he is not going to learn to hunt squirrels for subsistence. He needs kibble. And that's what we need to do with these T-Rex microbes, is take away their ability to go hunt their own kibble so that they're reliant on us. We need to domesticate them. That's how we ensure that they don't go rogue. Nobody's worried about golden retrievers going rogue.
What about mosquitoes, though? I mean, we've been hearing a lot about using CRISPR for gene drives for mosquitoes to limit mosquito-borne diseases.
We have to be really careful about, because we're not talking about they're domesticating the mosquito.
Right.
And that's the difference. The mosquitoes are not going to go rogue, but I also don't think that they're going to maintain a CRISPR system. One, it's still going to have the same problem it has in the human genome. It's going to be inaccurate, and therefore it's going to be anti-selected. It's going to be a chore. It's like asking a wild animal to go around with a brick around its neck. The first rock it can find to rub itself on to get that brick off, it's going to. The difference between domestication is you can control a population and make sure that, it's like, dude, I like that brick. You ever met a dog that gets upset when you take his collar off? Put the collar back on me. Yeah. That's how you want to do it. I'm a little leery of editing wild populations, not because I think they will go rogue or haywire or anything bad will happen. I just think it won't work.
Okay. When will I be able to inject bioluminescent and chromatophore genes into myself?
You can go ahead and do that right now. That is not going to work. But hey, write up a paper. Write up your observations for me. You might want to shift your focus of this research into, instead of injecting the genes into yourself, maybe getting your bespoke bacteria that's already on your skin to do it for you. That'll be a little easier. Let me know.
Are all controllable traits mutually exclusive? For example, the German Shepherds. Now do we know that the hip problems are not actually tied to loyalty?
Oh. So we'd have to have like limping buddies, and if they didn't limp, oh no. Loyalty is a very complex trait, and hip dysplasia is not as complex. So we think, especially now, there's dog genome sequencing projects, and dog owners are great about this. They're like, oh yeah, use my dog. Look it's much easier to get data, and the dogs aren't worried about privacy issues. So you take and we can check for this. So if that does end up being the case, that would be super interesting, and maybe we'll try to manage the hip dysplasia. But I suspect that it is not tied to loyalty, and we'll be able to tell in dogs that have it and don't have it exactly where the loci are, and we might be able to avoid those in the future.
Awesome. Well, thank you so much, Sarah. Thank you so much for the great questions, everybody.
Thank you.